Yeah, good afternoon, everyone. Welcome to April's A3D3 seminar. I'm very pleased to be able to introduce today's speaker, who's Professor Adam Charles. Adam is an assistant professor of biomedical engineering at Johns Hopkins University and has affiliations in the departments of neuroscience, the Center for Imaging Science, the Kavli Neuroscience Discovery Institute, and the Mathematical Institute for Data Science. His lab focuses on machine learning and signal processing for neural imaging, data analysis, and other applications. And these types of analysis and the lab require and develop tools based on probabilistic and low-dimensional modeling to create the next generation of methods that are needed to acquire and interpret complex neural signals. So we're very pleased to have Adam with us today. And we look forward to his talk on enabling big neuroscience through computational advances. Adam.
Thank you so much, and thanks for inviting me. It's I love talking about this stuff, but especially there's a couple aspects that I get to highlight to this particular audience I don't normally get to talk about, so this is going to be a lot of fun. So thanks again for the introduction, for the invitation, and okay, there we go. And as you mentioned, right, my lab focuses on this, interface of data acquisition, data modeling, and how we can leverage the statistical structure of data, both in the acquisition process and the data modeling parts of our process of creating scientific understanding through observation. So just to kind of give you a high level, I sit at this interface, and I love that there's so much additional progress being made on bringing this combination of theory, application, data science, and neuroscience kind of all together. And it really speaks to what the kinds of questions that I love to think about, which is how do we create new neural recording technologies? How do we model how neurons represent information? And finally, closing the loop, how do we use those inspirations of the challenges that arise in neuroscience, both in our learning of how the brain operates and computes, and just learning from the challenges of understanding how the brain computes and thinking at different scales and different types of data and types of scientific questions, how to create the next generation of algorithmic tools. So I want to start today by talking about the acquisition part of my lab. And in particular, I'm going to focus on optical imaging. So calcium imaging is going to play a big role, but actually I'm going to talk today a little bit about optical imaging in general. Optical imaging is an alternate way of seeing the brain from electrophysiology. Electrophysiology has a lot of advantages, but optical imaging is there to complement the powers of electrical imaging to try to get at addressing a couple key issues that we want to understand about the brain. So one aspect is we are quite a few orders of magnitude away from whole brain recordings. Even the best tools that I could think of that operate at a neuronal scale, right? So imaging or recording single cells and populations thereof simultaneously, we are quite far away from even capturing one entire brain area, let alone the whole thing. The second is that there's incredibly important details at a variety of scales. There's importance in the synaptic activity that goes on and the adaptation. There's importance in the dendritic activity that propagates. There's importance in the somatic activity and how spikes are propagated down. And there's important activity at the group level where populations of neurons co-activate in order to create computation. And this computation is local, but it's also global. And so we both need to think about how these populations interact from inside a small population, as well as across between different circuits that represent different inputs and outputs of our system. And the last thing is that there's a lot of individuality. And so we really have to be able to account for the variability in the computations and how different individuals in different situations might create additional complexity in how we interpret this data. So I'll start with optical imaging here, and just to kind of show you how far we've come. This was a video that I used to show back when I was on the job market. And so this was pretty state-of-the-art back then, right? It's a small mouse running on a ball in a virtual environment. So there's actually a curved screen around this mouse. And as it's running around, right, there's a feedback loop that creates this virtual environment that it can navigate. It learns to navigate. And because it's head fixed and we have this microscope attached, we can optically record from inside the mouse's brain. And so here is a 500 by 500 micrometer plane recorded in retrosplenial cortex. And this is time locked to the video. So as you see the mouse running down this hallway, it's doing a decision-making task where it has to count the number of pillars on the left and right. You'll see them kind of show up. And it has to go left or right based on which side had the most pillars. And so this was data taken by Sue-Ann Coe, now a group leader at Janelia. And really, you can start seeing that each of these flashes of light, which represent individual neurons activating in the brain, can be synced to the video and can be synced to the behavior to understand how this computation is arising in this particular case, decision making. This was back then. Here's a slightly newer version of this whole thing. So this is from Bijan Pesseron's lab at UPenn, who I've been collaborating with. And he's been working on optical imaging in macaque. And so we've gone to much larger species with much more area to cover, much bigger brains. And we're still taking these images that are, 500 by 500 microns, and we're able now to navigate around this extremely large area, which requires all kinds of new tools in order to isolate and identify where the right spot is to record, or how do we make sense of this new signals that have just fundamentally different characteristics than similar looking signals do in mice. So just kind of a primer in case anyone has not had the opportunity to deal with multi-photon imaging in the past. The types of imaging that I've been working the most with is 2-photon imaging, where part of the benefit is, as opposed to typical 1-photon imaging, where you shine a light and you use a CCD array to capture an entire image at once. In order to penetrate deeper, you use a lower energy light and you pick a scan depth under the tissue, and you actually focus your laser into that depth, illuminating one point at a time and raster scanning back and forth through the volume. And every cell that you happen to hit fluoresces. You get an image of that cell in the picture, in the frame of the video that you get. One of the nice things is, because it is focused into a particular depth, out of plane cells don't affect your measurement as much as they would have if it was a 1-photon imaging system. And so you can select a population of cells by changing that focal plane and moving around and focusing on where it is that the signal really appears. So things that we want to do with this data has extremely rich information. It is full spatiotemporal videos of fluorescing components, and that fluorescence, those deviations, those wiggles of fluorescence are indicative of neuronal firing. And so we might want to extract out each cell, and in this case, we're going to call them spatial profiles, and their individual time courses, so each of their fluorescent activity over time. In particular, most of the time, we're interested in these bursts of activity that represent bursts of spikes that occur all at once. So there's a lot that we want to do with fluorescence imaging. We want to increase the quantity of the neurons that we record. We want to see more of the brain in order to understand the computation in a more complete way. We want to increase the quality of the processing algorithms. We want to image across different scales, right? There's rich information at a really multi-scale in the brain. We want speed, right? So there's a lot of promise, and I think this is a great crowd for this, right? There's a lot of promise in being able to do real-time analysis, online experiments, providing feedback and perturbations to try to suss out causality rather than post hoc analysis. And so speed is really of the essence. And assessment is also incredibly difficult in these cases because oftentimes this is the only avenue we have in which to see the data in this way. We cannot look at the same spatiotemporal components in any other way. And so we need to devise new ways to assess. I've had the wonderful opportunity to try to tackle problems across scale. And I'll isolate just a couple of these to highlight, the main points of where the field is and where at least my lab is trying to take it. So I want to start with quality. And this is actually a little different than calcium. This is a much more difficult scale to work with. This was a project where we wanted to zoom in so much that we could image individual synapses and track those synapses over time. And what I mean by that is we partnered with Rick Huganier, Dwight Burgles here at Hopkins, and Tiger Xu is a grad student who really pushed this project in a very amazing way, and collaborating with Austin Graves and Gabby Kost. Austin and Rick had developed this indicator that actually tags not calcium-based fluorescence, but it tags individual AMPA receptors. Moreover, it's pH sensitive. So it only fluoresces if it's at the cell membrane, not inside the cell. And so what this means is that if you image, you will see bright spots only where there are significant AMPA receptors at the membrane, meaning that those are AMPA receptors that are actually responsible for the glutamate and sodium movement through into and out of the cell. And so this is incredibly powerful, right? So being able to see the receptor at the surface, right? That's this little picture up at the top right, means that we could get these images. So at the top here is a SEP-GLUA-A2. That's the name of this indicator that fluoresces. And each of these little dots is a single synapse, right? So it's a single spine with AMPA receptors. And the temporal aspect of this is an anatomical, right, structural image, but it's completely in vivo. And so what that means is that you can come back the next day and the next day and the next day, and you can see what happened to those AMPA receptors. Did they change or are they the same? And that lets us track at scale synaptic plasticity, essentially. So what is the challenge here? The challenge is that these are incredibly small objects that we want to track. We want to be able to track them day to day so that we can assess where learning or adaptation has happened, potentially perturb the setup if we want to try to test different hypotheses. Except, what we see is this image right here in the middle. Incredibly blurred, incredibly noisy, just very low SNR, because just the ability to get high quality data at that scale in vivo is incredibly hard. You could try to do something like high resolution or super resolution area scan imaging that works great in vitro, right? So if you have a little dish and you have your sample in the dish, it works beautifully. Look at these like beautifully isolated synapses, right? It's amazing. It does not work in vivo. Slight motion just ruins the whole thing. And so we really wanted the resolution of area scan. We wanted the penetration and ability to use it in vivo that we get with 2-photon microscopy. And so we used machine learning to create this in between what we called cross-trained care. So what is this cross-trained care? What we did was we extracted slices of tissue that had been introduced with this indicator. We imaged them ex vivo, both with ARIScan super resolution and one photon imaging, right? So one photon is blurrier than two photon. You don't get that nice sharper resolution. So it mimics the quality that we expect to have in vivo with two photon. This allowed us to create the ideal training set to train a neural network, in our particular case, a UNet, based off of the CARE algorithm that was developed and released a few years prior. And we're able to then, given in vivo two photon data, feed it into the same network as if it was this blurry one photon data. And just like this network was trained to predict the super resolved tissue from the one photon, it then created a super resolved version, a nice cleaner version of the two photon using the learned statistics of this type of data. So we tested this quite extensively. I am someone who is very terrified of machine learning having a bias problem and overfitting and hallucinating things. We tested this in a myriad of ways, including, feeding in dendritic data and seeing if it broke it up into little pieces of circles, because it was basically trained on small circles. It passed, so we went forward with releasing the code. And here's just a handful of comparisons showing the benefit over other types of denoising algorithms, including standard things like non-local means, deconvolution, BM3D, which is another kind of more state-of-the-art version, and noise to void. Right? These are all methods that you can find in the literature. They've been used. They do help. They do make this raw two-photon data better quality. But this cross-trained care just seemed to get overall higher PSNR and lower normalized root mean squared error. And so we were able to really clean the data to a level that we were quite happy with. This is a little video that I like to show. So just to show the, this is to show that this is really this is volumetric data. So everything I showed you was images because we're kind of stuck with that for static visuals. But for dynamic visuals, like this video, you can really see that this is a whole volume that we're imaging with hundreds of thousands of synapses in there. So what can we do with this? We so far have been using this for a number of different scientific applications. Rick's lab is really pushing forward a number of these. Here's an example of fear conditioning. Oh, sorry, no, this is the enriched environment. My bad, that's a different project. So we let the mouse just kind of hang out in its home cage for a number of days. And then on day 4, we put it in an enriched environment with all kinds of new stuff for it to learn and adapt to. And the hypothesis was that we should see a significant amount of additional synaptic plasticity changes, right, as depicted by the AMPA receptors that we're imaging after this day 4. What we didn't expect is that when we kind of circled and labeled and tracked these synapses that we can now identify one at a time, almost 50% of them were changing in the home cage environment before any additional enrichment. After enrichment, a whole lot more of the AMPA receptors were changing. It was a much more dynamic change in the network. However, right, even in this home cage environment, there was a lot of synaptic plasticity going on. And so that we thought was really interesting. And, as we get these algorithms faster and, working on a day-to-day basis, we could think of how to use this information to test what those synaptic changes are doing when the animal is, just living its normal life, right? Not doing anything particularly different than what it's been doing for the many days prior since it got the cranial window installed. We're also kind of going further with this. So in a collaboration with the Allen Institute, we're using new optics to try to image not just the anatomical strength of the number of AMP receptors, but the actual glutamate release using some fancy new optics developed by Kasper Podrowski, the SLAP2 sensor. And so this is a sensor that really focuses on individual pieces of light rather than taking whole images. And we've been able to do some really cool things there, like online registration to a volume, which lets us do online motion correction for this type of data. All right, so part two, and I wanna go through this a little quickly because I want to make sure to get to the last bit, which I think will be perhaps very interesting to this particular crowd. But I do want to mention, right, in this collaboration with Carl Mischne, we started looking at the variability in the types of data that people are collecting and what we can do with this algorithmically. How can data science allow us to treat the different scales of imaging that is being done? In particular, most of the imaging that has been done so far has focused on soma, right? Somatic imaging, cell bodies. They're these beautiful little donut shapes because the nucleus doesn't really get any fluorescence. And so you get these beautiful shapes. You can do all kinds of things to try to identify what those circles are. But if you zoom in a bit, you get dendrites, you get spines, right? This is very different morphological features. If you want to develop algorithms for these, you can't use the same exact constraints and regularizations that you might use for cell bodies. And wide field, go figure. I don't even know that we know that there are very specific boundaries to the objects that we're expecting to detect in wide field. We very often see correlated activity across boundaries of atlases and, constrained to within subsets of these boundaries of atlases. So the atlases that we have of brain-wide spatial distribution of activity are really mostly a guideline for this type of data. So, right, to back up a little bit, right, we're talking about the problem of extracting the individual fluorescent components. Often this is phrased as a matrix factorization approach, right? So you want to find your spatial profiles and time courses A and Phi, such that when you multiply them, right, so you have a neuron by component, or sorry, a pixel by component and a time by component matrix, and you want to multiply these two together to recreate the video. And you have all kinds of regularization that people use. And there's been a lot of really great work on making these types of algorithms really efficient. They all make assumptions, right? This regularization are things like spatial cohesion or, they look good as per a trained CNN. Things like that are built into this R function here. We took a slightly different approach. We said, okay, well, we're gonna just back off of the whole pixel space idea. This was great for somas. Pixels nearby in space were very likely to belong to the same soma if they did belong to any soma. But in dendritic and wide field imaging, we don't have this assumption anymore. So what we did was we went with this graph signal processing approach where we used the temporal correlations of each pixel. So here there's a blue pixel, there's another blue pixel. They're very correlated. They have the same time trace. This yellow pixel is much closer, but it's a different cell with a completely different time trace. And this green pixel is actually the overlap of the both of them. And so what ends up happening is that if you map all of these to a graph based on their temporal correlations, pixels that are nearby on this graph, so each node in this graph represents a single pixel. The graph is imbued with a temporal direction, right? So this pixel has a time course. It was the original pixel's time course that mapped onto it. Pixels nearby on the graph are more likely to belong to the same component. And so we can use that information to take very, very distributed activity of components that are not nicely co-located and identify them very easily. So we call this graph filtered time trace dictionary learning. It is essentially a map procedure done through dictionary learning. So I won't go into too many of the details. Suffice it to say that this is an alternating variational minimization, much like the original dictionary learning procedure was from Olshausen and Field and Mickey Alad and others. What I will go into is how we implement the graph. And the way that we do is we assume that each pixel, so Y represents here the time course of a single pixel. A represents how much of each of the time traces that you're going to learn contribute to this pixel's activity. So let's say there were 500 components in your field of view. This pixel is going to have a time trace that is composed of some number of them. And due to the fact that that slice of tissue that you're imaging and sectioning optically is very small, or at least very thin, if you have good optics and you aligned everything right, there's not very many of those objects overlapping on any given pixel. And so if phi is the set of all time traces, A is saying how many of those time traces create each pixel. Phi A is going to have a sparse representation because most of the A's are going to be 0 for most of the pixels. Mathematically, we can write that as a Laplacian prior, where in our case, because the A's are going to be all positive because fluorescence never goes negative, this is actually going to be only an exponential because it's going to be conditioned on A greater than 0. And what we do is we actually create this hierarchical model where we add one more step up. And this step up is saying that the variance of this Laplacian distribution is going to be dependent on its neighbors on the graph. So in particular, we pick a gamma distribution. Gammas are conjugate to Laplace and exponential, so it makes the math work out. But practically, it's also very nice, and that it gives a very direct way of saying, okay, if A is on for pixel 1, right? If a particular time trace contributes to pixel 1, then we want it to also have a representation of pixel 2 that looks kind of like that. And so kind of, waving our hands and saying, hey, we're going to do this across the entire graph and just kind of share information across the edges of the graph, which are given by the temporal correlations. What that yields is this reweighted algorithm where every time that we want to identify A, the contributions of each time course to each pixel, we first solve a lasso, except we don't solve a typical lasso, right? Typical lasso would tell us just, hey, this is going to be sparse. We solve a lasso with this gamma, lambda. I promise I know my Greek. This lambda that will weigh each possible time trace that could have contributed to your pixel differently. A larger value will make it harder to attribute activity for that pixel to that time trace. A low value will mean it's very likely that that particular time trace appeared in the pixel and that component was there. And so this is a way of modulating this procedure. And so we go back and forth between doing this weighted lasso and updating these lambdas where there's a divisive term down here that depends on the average value of the activation on all your neighbors. So all your neighbors are active. This lambda gets really low. And it's very likely that that pixel will also have the same representation in component space. All right. So we tested this algorithm in a number of different ways. So first, we used actually a simulator that we developed to create fake data, basically, to create very highly realistic two-photon calcium data. And what we found was that we did quite well, right? So, you know, the typical algorithms that people use are the CNMF K-min and SWE2P. They found in this data set about 300 independent unique components. We found about 435-ish, I think, 36. Interesting to note, despite their prominence in many other places, and you know, I use them as well, deep learning-based methods like ST-neuronet did not perform very well. And this gets to a really interesting thing that we can talk about, which is it's great to amortize your learning so that you can run these neural nets online very quickly. They don't seem to extend easily to other labs. There's this out of distribution problem that shows up. And, you know, they essentially end up overtraining a bit to the type of data that you're dealing with. And so I've seen this in a number of ways. I've seen this here with cell finding. I've seen this on denoising algorithms, that if you take a denoising algorithm trained on mouse data and you try to run it on macaque data, all of a sudden you need to do all of these fine-tuning and update iterations in order to embed the new statistics into the data. And this could be as bad as you try to give your algorithm to the lab down the hall that's using a different microscope with slightly different offsets of the optics and slightly different surgery preps. It's very insidious. And you know, if you'd like to, Gal and I wrote a little thing on this that we published on the archive just 2 months ago as part of neurophotonics. So if you're curious about those kinds of things, go ahead. I would love if these things worked better, but sadly, it seems like every lab is out of distribution somehow. More importantly, we tested this, and I'll just to make sure that we have time, I want to jump to the dendritic components, right? So we took some data from our collaborator, Jackie Schiller. They imaged dendrites and motor cortex. And so here is a sparsely labeled data set. So only 6 total dendritic arbors were in this particular volume that we had. It was nicely annotated anatomically, so we had some semblance of ground truth, even though we didn't have the temporal ground truth, we had the spatial ground truth. And what we thought was very interesting was that our algorithm found this dendritic arbor, but split it into 2 different components. We were a little curious. I mean, we were already thinking, okay, this is at least better. You know, SWE2P broke this up into about 200. So, you know, that's better. We were curious about this. Why did it break it up into 2 components? It turns out that this is actually a z-drift issue. So the plane of imaging slowly shifted down as you are imaging. And this is, you know, for example, in the previous project, when I mentioned dynamic online compensation for motion, this can be extremely difficult to compensate with offline once you've already collected the data and are dealing with it. It's a lot easier to, you know, refocus the microscope online if you're having these kinds of issues. And so in our particular case, how this showed up is we ended up with the shallow green and the deep blue component that were, you know, offset just a couple microns from each other, but still were enough to show up as 2 different objects. But still better than 200. And in fact, you know, this is something that we saw a bunch, right? So of the 6 cells, it seemed like only 3 of them had any temporal activity. I will say, like many algorithms in this space, our algorithm is blind to anything that doesn't have activity, right? It's kind of like a T-Rex. If it doesn't move, we don't see it. And that's because it depends on these fluctuations over time. And yes, we do break up the 3 cells that we see into 2 or 3 components because of the z-drift. But if you look at SWE2P, they take those same 3 components and they break it up into about 200 each. And so the stitching problem of putting those back together post hoc is much harder. Here's an image that I particularly like, which is Jackie and Nate then gave us a much denser volume to work with. This is a lot of components all bundled together. So it's like the spaghetti problem where you have to disentangle all these different wires that are each a separate dendrite. And we do quite well. So here's a couple examples where despite these dendrites kind of being all over the place, we can identify them, identify their time courses, and start thinking about, what is it that they're doing. One interesting thing is that some of these components seem to be found twice, and we were a little confused about that. Here's a blue dendrite, and here's a pink dendrite. And they look like they're the same object here, maybe a little bit of extra stuff on this pink. Maybe the fluorescence distribution is a little different. And when we dug into this, we noticed that, yes, there are times where these are not linear combinations of each other, right? These are actually, different in their activity profiles. And sometimes you get quite a big bump of blue and not a big bump of orange. And if you look in the video, you actually don't quite see the purple things show up, right? These extra pieces that we saw that the purple had. And the other thing happens as well, where sometimes the purple's really bright and the blue's not really there. And so this is giving us hope that these types of algorithms that can really disentangle very subtle differences in signal can help us learn about things like dendritic processing by segmenting the activity even within a single dendrite. So I'll skip over the wide field. I'll say that it does work, and we can find meaningful components. I do want to pause for a second and say that we are really trying to get this tool out into the community. A very talented undergrad, Alex Estrada, helped develop a GUI for this particular algorithm. So, right, we can not only run the algorithm much more easily. It's compiled so that even if you don't have a MATLAB license, you can use it by just using the standalone app. And it has a couple extra features of being able to go back and assess the decomposition that you had, look at the data in different ways, and try to identify how well the algorithm has been doing on your data. Similarly, a very talented undergrad in my lab who is just graduated, she'll be starting grad school at MIT next year. Gabby Kang has been working on some compressive matrix factorization tools to make this all run faster. And I wanna keep that in mind for a minute. I'll come back to that in one slide. We've also run it on other data sets. So here's functional ultrasound, right? There was nothing in the assumptions that relied specifically on the calcium signal properties. And so we could take the same algorithm, and we've been using it for functional ultrasound imaging, in this case, in a freely moving rat. So this is a rat that's completely just not head fixed, running around, self-initiated decision-making tasks. And we can get extremely stable hemodynamic activity, in this particular case, from the surface of the brain all the way down to the brainstem. So there's a bunch of great tools coming out for this, and I'm very excited about functional ultrasound. But first, I want to go back for a second, right? So there's a lot to do in calcium imaging. As I mentioned, I've done a bunch, right? So stereoscopic imaging, robust processing, simulation-based assessment. But I want to talk about this online problem because of this crowd. This is new work, so this is my first time talking about this. Can we do real-time stuff with calcium? And up until now, the answer has been no, because you have to get the data off the animal. Well, really, you have to get the recordings, you have to get them, you have to do this huge matrix factorization problem, solve it, assess it, probably rerun it 2 or 3 times to choose the right parameters. There's no time to do this inference adaptation loop. So what we've been working on is a method. So there's been, I would say this is perhaps the 3rd attempt in the literature that I know of. The other 2 being on acid from the Cayman makers, and then Fiola, which is a GPU-based method. We wanted a lightweight CPU-based method that could online identify cells and extract their activity in real time. And what does real time mean here? Data is usually obtained at a 30 hertz rate. So you have to process every frame that comes in. True real time is you process every frame that comes in within 1/30 seconds, and possibly faster to leave time for all of the adaptation and feedback and all of that fun stuff that you need to do. So this was kind of daunting because up until now, right, it's like, here's a video that took half an hour to collect and run it for 3 hours on, whatever hard drive or whatever hardware that you have. Not fun. Yulia, an amazing undergrad, lots of great undergrads. I've been very lucky. Was working with me to take an algorithm that we had previously worked on for robust inference and bring it to the online setting. And this is the heart of the algorithm, is this pseudo algorithm. It was published about 1.5 years ago in Nature Methods. It focused more on the offline setting where you want to make sure that activity from one cell does not bleed into another cell. In real-time imaging, you don't know where the cells are. So this is a real problem. If you average a bunch of pixels that you don't know if it overlaps with cells that you have no idea that they exist yet because they've never fired before, you get a lot of false activity. And we want to avoid that. So we use this as this heart of our algorithm where frame by frame, as data comes in, we run it through, given the dictionary of cells that we know about, and then use the residuals to update the dictionary if we detect something new. It works. I apologize. I don't know why this is so small. I'll just summarize, right? If you can see here, as the algorithm goes, these cells that are found in order, in temporal order, right? This is an order. And you can see that as they're found, the first firing activity is later and later, right? So it's finding these in order that they first become active. We compared against OnAcid, which is kind of the version that existed so far. And what we found was we were much more robustly faster, right? We were running at approximately 80 to 100 hertz as opposed to their 20 to 30. And as I mentioned, right, 30 hertz is already borderline because you still need to do all the other processing steps. And so this is submitted. Hopefully we'll release the bioarchive and the code. This required a lot of fancy C++ to get this to work properly on a CPU. But we wanted it to be available even to people who didn't have a GPU. All right, so last bit. Okay, great. I've got a little bit of time to get through this. The last bit, I want to take a small jump from the first half of the talk, which was all about how we get the data, process it, and produce some sort of really raw neural representation of the data, whether that be, AMPA receptor level, whether that be glutamate activity or calcium activity, something that is just kind of like a raw measurement of the data, nothing about like latent spaces or dimensionality reduction or anything like that played in. So that's here. And this project actually is very apt because the paper just came online today at Journal of Machine Learning Research. So you can go find it there if you're curious, or hopefully I do a good job of explaining it. And the whole goal of this particular project was to create a model that could tackle a couple very core challenges in trying to interpret functional neural activity as a dynamical system. So to start with, right, let's say we believe two things. One of which is that neural data is a dynamical system. I think this is the least of the assumptions that we can nitpick on, because there's very known causal relationships where neurons influence other neurons. And that happens over time. The other, which is, I think, a more becoming more established over time is that there's this neural manifold, right? The activity of data as defined by, hey, let's string up our activity of all our neurons in our population into a state vector, right? Where element 1 is neuron 1, element 2 is neuron 2. That does not take up the entirety of the space, right? If you follow the trajectories in time, they lie on this low-dimensional but nonlinear surface. Where the surface comes from, we can debate. All I'm asking right now is if you do believe in it, that's great. If you don't, suspension of disbelief for a second. I'm going to assume very little about that surface other than it exists. And furthermore, we could go a step further and say that wherever this surface is, it's not directly related to the data, or sorry, it's not directly the data itself, but you could think of it as being in this latent dimensional space where the actual data Y is some readout of this latent dimension, right? In this case, in this model, we're going to assume it's linear. I'm going to assume it's linear because I want my dynamics to be nonlinear. And there's a identifiability problem if both your readout is nonlinear and your dynamics is nonlinear. You could basically have any type of diffeomorphic changes to one and compensate for them in the other. It becomes a whole mess of identifiability. So here we're going to assume that the readout is linear. So why I'm bringing this up is if we believe these two things, then the dynamics itself, when it moves you along the manifold, cannot move you arbitrarily in neural space. It can only move you in directions, in actions that keep you on the manifold. Otherwise, you would not have a manifold. That's all I'm going to assume. And in particular, what this gives us is it gives us a really powerful way to talk about dynamics in the tangent space, right? So there's a tangent space to this manifold that defines, in essence, all the ways you can go locally in a very small area on this manifold that keeps you there. It doesn't go orthogonal. It doesn't like push you off the manifold, right? We're assuming that that's not an intrinsic part of the system. If that happens, it's because someone came in with a bunch of electrodes and shocked the brain. Or I guess optogenetics now. We're getting fancy. So I think that this points to something that has bothered me, which is if you look at the space of dynamical systems that have been applied to neuroscience, they're either linear dynamical systems, which have gotten us very far and have been very inspirational, but essentially treat the entire system as one linear system, right? So very interpretable in terms of the interactions of cells with each other, not very expressive in terms of the amount of the systems it can capture. It's not non-stationary, it's not nonlinear. There's RNNs, right? There's completely nonlinear systems that could do arbitrary things. They're extremely expressive. We've learned how to fit them to data. Amazing. They're not very interpretable. You have to do a lot of extra work after you fit the RNN to extract meaning out of that system. There's been extensions, right? So if you want to go towards non-stationarity and approximating nonlinearity with non-stationarity, switched linear dynamical systems are a really great model that has been like revitalized by people like Scott Linderman. He's created a lot of amazing tools for us to use this and you know, shown us what it can do, right? These types of systems, you have a linear system that switches every so often. So you might start with system 1 and jump to system 2, then jump to system 3. There's a limit to the expressivity of this, but there's one thing that retains, which is at every single time point, the entire system is modeled with a single dynamical system. That bugs, right? That is all of these systems, by the way, right? And how that bothers me is that if you think of the brain, it's incredibly distributed. It's highly parallelized. We have lots of little pieces operating, you know, not 100% independently, but quite independently. And if you want to identify what one system is doing independent of another, you need a way to be able to, you need a language in order to talk about their combined activity changing over time and how they can contribute differently in a compositional way. So our model, following the tangent space idea, says, okay, well, those different things, that compositional way, what we mean by that is that our action, our A, our matrix, our linear dynamical system at time t is going to be a combination of some known dynamics F, which are going to be essentially the directions on this tangent space. And at every time t, we're going to take a bunch of coefficients C, and we're going to build up our dynamical system, our linear dynamical system. So we retain the interpretability of linear dynamics, but we're going to build it out of pieces at every time step t that we're going to learn from the data. I put this up because we do have a continuous time version of the model, but I won't talk about that much today. But we can do this all in continuous time as well. I'll just focus on the discrete time because it's what we've been working with the most for a number of computational reasons. So model-wise, right, we could write this as the following system, right? So x of t at time t minus 1, this latent state underlying the neural data, generates the neural data, and through F creates the next latent state, right? So you should be having some flashbacks to your Kalman filter time and whichever class you happen to see that in. And the other thing is that you have this higher level set of coefficients C that also tell you which Fs to use at time t for xt minus 1 to generate xt. And this keeps going on and on and on and on and on. Now, this gives us an option of how to model these coefficients C, right? If we think that the C's are going to be a vector of, let's say, a 1 at one time point and 0 everywhere else, that recovers a switched linear style system. What we do is to imbue this with interpretability. We say that C's don't have to be just one on at a time, but we don't want all of them on at any given time, or we wouldn't be able to correlate what do the dynamics mean in terms of, let's say, the task, or in terms of what the animal is seeing. And so we choose sparsity. So the C's are going to be sparse at every time point t. Fitting this boils down to a maximum a posteriori algorithm. We only want to find D and F, right? We want to fit the model parameters given the data. This creates a expectation maximization style algorithm. I'll skip over the details. I'm happy to talk about them if anyone's curious as to the derivation and so forth. But I do want to point to one thing, which is there is an inference step here that's quite costly. And that inference step is finding, right, somewhere in the middle, we have to estimate the state and the dynamics state, so X and C, at every time point t throughout our trajectory. This gets really hard because both X and C are sparse, and so this becomes a really difficult problem. Moreover, there's this product of X and C that shows up in the optimization. Now, something that I think will be fun, if anyone has thoughts or ideas in this particular group, is that the way that we can solve this efficiently and quickly is actually to turn it into an online algorithm. And so I had done, luckily enough, a bunch of work early on in my career trying to devise Kalman filter-esque type algorithms for sparse data. It turns out that's exactly what we need here, right? We could rewrite this as an algorithm, as an optimization, where given the data at time t, we're finding both the state at time t and the dynamics that took us to the state at time t, conditioned on the previous estimate of the state. And we can run this forward in time, and it turns out, don't read the details of the proof here. This is just to say we can prove that it converges to a steady state error that is the noise floor. This is what we did back in the day when we were studying these types of systems. relies on compressed sensing type of technology, but just to say that this is the right thing to do and it works. All right, so in the last few minutes, I have 1 minute, I think. Can I have 2?
Sure.
Yes. All right, thanks. So I just want to highlight a couple key points of using this thing. One is that the compositionality works. So here's a test that if this failed, I would have probably not really been as excited. I might, we created synthetic data of 5 red neurons and 5 blue neurons. We ran a system forward where the 5 red neurons switched between 3 different systems, right? So the white bar here tells you which system is currently running the red neurons. Similarly for the blue, each system is independent and they're switching at different times and they have different dynamics. And we wanted to know, could we learn the compositionality of this? Or is it the case that what we're going to learn is only that combinations of the 2 systems operating together, right? This is the whole reason why I, wanted to advance beyond what has currently been the state of the art. So we even gave the algorithm plenty of rope to do whatever it wanted. We let it learn up to 15 different dynamical systems. We said, go for it. You can so easily overfit. And it turns out that when we ran our algorithm, it did in fact find this compositional structure. It found 3 systems that switched off with the red. It found 3 systems that switched off with the blue, and the rest of the systems were not used at all. And so it found the right thing. We also ran this on a couple other tests. So here is a Fitsunugumu model. And where this was showing was the effect of sparsity. So if this is a simple model of a neuron where there's a nice depolarization stage and you can see the state space U and V and W that dictate this dynamical system model. What I want you to focus on is this column all the way on the right, where C1 and C2, if we turn off the sparsity, learns a representation of the data, but it's all jumbled up. Meanwhile, if we induce sparsity, we get this beautiful separation of low and high voltage states in C1 and C2 separately. And so we've learned something significant about the way that this model operates by inducing sparsity. Finally, we've been running this on zebrafish, not zebrafish, C. elegans, zebrafish is next, and mouse. So we're learning connectivity in mouse cortex. This is data from the IBL. And we're really trying to push this forward in the sense of learning really brain-wide dynamics with, trying to meet the challenge of this beautiful data that's being created that is capturing so many different areas and so many different systems simultaneously, and we need to know what they're doing. So with that, I'd like to thank all of my collaborators. Right, so Tiger, who really pushed forward the XTC, Gal Mishna, who was my partner for our the graph algorithm, Noga and Eva and Yenho, who worked a bunch on the dynamical systems work and are pushing it forward into different neuroscience directions. And of course, I'd love to thank my funding, NIH, Chan Zuckerberg, Kavli, and the Johns Hopkins Discovery Awards. So with that, I'll leave you with some references to the papers of the work that I've discussed, and I'd love to take any questions now by email, show up at my office, wherever.
Great. Okay. Thank you, Adam, for a very interesting talk. I think we've lost a number of people as we're past the hour now. So for those who have managed to stay on, let's see if there are 1 or 2 questions. Well, I encourage any of our remote viewers or later viewers to contact Adam if they have questions on this very interesting material. And I thank you again, Adam. Yep. So
Yeah,
I think
well,
we can
thank you
stop
so much.
recording, Glue.
Yep.